Assalamu alaikum everyone and welcome back to my channel. This is my second video in this series of videos that I've started making, and this is another research paper that I published quite a while ago, so I've decided to present it to you. So let's get started. The title of my PowerPoint is Chitinase 3-like protein 1 in health and disease. So let's start with it. Okay, so chitinase 3-like protein 1 is also known as glycoprotein 39, breast regressing protein 39, 38 kDa heparin binding glycoprotein, Conrex 40 kDa, and mammary gland protein. So it was initially discovered in the culture supernatant of fetal osteoblast cell lines, and it is a glycoprotein. It weighs 40 kDa and it belongs to the glycosyl hydrolase family. It binds to chitin, heparin, and hyaluronic acid. So in non-human chitinases, it catalyzes the hydrolysis of chitin, which is a polymer, to its monomer, N-acetyl D-glucosamine. So it has no chitinase activity in humans because it has leucine instead of glutamine in its active state. So when is it expressed in the body? Well, it's not expressed under physiological conditions, but it is observed in patients with inflammatory diseases. Now, those inflammatory diseases include rheumatoid arthritis, osteoarthritis, inflammatory bowel disease, sarcoidosis, chronic obstructive pulmonary disease, asthma, atherosclerosis, type 1 and type 2 diabetes, liver fibrosis, and a variety of solid carcinomas. So we're going to focus on liver fibrosis in this paper, okay? So this is the structure of chitinase like 1 protein. There's no rocket science to it. It's a glycoprotein, right? So you can see all these structures that are, this is the protein portion, and this yellow portion in the middle is the carbohydrate portion. So it's a glycoprotein, right? So it's made of carbohydrates and proteins. And this domain that you see is the domain where the chitin binds to it, okay? Okay, this slide is showing you its functions. So it has a chitinase function in plants, in bacteria, in fungi, in insects, but it does not possess this chitinase function in humans. So what is the chitinase function? It breaks down this polymer, which is chitin. It breaks it down into its monomers. So this activity is absent in humans, but it's present in these organisms, okay? Okay, so previously its role was unclear, but it was expressed and secreted by a very diverse kind of cells, such as macrophages, chondrocytes, fibroblasts-like synovial cells, vascular smooth muscle cells, and hepatic stellate cells. It is also expressed in inflammation and tissue remodeling and fibrosis. So prior research has proven chitinase 3-like protein 1 antigens in areas with active fibrosis in immunohistochemical analysis. Several studies have established that this is a biomarker for fibrosis induced by alcohol and viral hepatitis. So if you feel like I'm going a bit too quickly for you, I have given the references of all the articles that I've used in this presentation, all the diagrams, etc. And it's all in the description of the video. So you don't need to worry about it and you can just find it in the description, okay? Okay, so this looks like a very scary diagram, but it's not scary at all, okay? So this is basically showing you the pathogenesis of liver fibrosis and how it starts. So there is a group of cells which are the culprits of liver fibrosis, and they are called myofibroblasts, as you can see over here. So these myofibroblasts are characterized by different kinds of cells, but most commonly they are hepatic stellate cells and portal sinusoidal cells. Okay, so what they do is these cells, they undergo an epithelial to mesenchymal transition, and this transition occurs in response to insult and injury. So when these cells undergo all these repeated insults, they undergo this transition, and as a result of this transition, they express asthma. Asthma is a smooth muscle actin, and it secretes extracellular matrix, type 1 and type 3 collagen. This results in this huge pool of myofibroblasts, and this pool of myofibroblasts, they encourage and they cause fibrosis in the liver. So this is the whole pathogenesis. These cells, there are two culprits, main culprits, stellate cells and portal sinusoidal cells. So when there is repeated insult and injury to the liver, they differentiate into myofibroblasts. Then there is a whole pool of myofibroblast cells, and these cells encourage the liver to fibrose. Okay, so let's move on to the next slide. Okay, so this is showing you the natural progression of liver fibrosis and liver cirrhosis. So let's start at the very beginning. This is a normal liver. Okay, this is the normal liver of an healthy individual. Okay, so when this healthy individual, for example, does not take care of his her liver, indulges in, bad health practices, or this person has dyslipidemia, has metabolic syndrome, has obesity, has diabetes, or any other lifestyle changes, this person may develop NAFLD. So what is NAFLD? NAFLD is non-alcoholic fatty liver disease. So from a normal liver, you can go to the first stage of cirrhosis, which is NAFLD. So what does NAFLD mean? It means that more than 5% of your liver now has steatohepatitis. So when more than 5% of your liver has steatohepatitis, which is also called fatty liver, then you have NAFLD, and it could be due to any cause. Now, what is the difference between NAFLD and NASH? Both of them have steatohepatitis. But the differentiating factor is that while in NAFLD you have steatohepatitis, there is no inflammation. But when there is inflammation in addition to steatohepatitis, then we label it as NASH. So you could say that this is a progression of NAFLD. Okay. And this NASH then progresses to advanced fibrosis if the injury and insult continue. Okay, till this point, this is still reversible. The damage to the liver is reversible to some extent if there are lifestyle changes, if there are interventions, the fibrosis at this stage can be halted or slowed down. But when the patient does not undertake any lifestyle changes, this advanced fibrosis can result in cirrhosis. So what is cirrhosis? Cirrhosis is when there is widespread distortion of normal hepatic architecture. There is irreversible fibrosis of the liver, and so much of the area of the liver is involved in this fibrosis that there is decompensation. I'll repeat it one more time. There is widespread distortion of the normal liver architecture. There is irreversible fibrosis, and then there is decompensation in the liver. Decompensation means that the normal functions the liver was carrying out prior to this injury and fibrosis, there is not enough viable liver tissue left that it's gonna carry out those normal functions, and now we call it a cirrhotic liver or a decompensated liver. So this is a very dangerous stage because from cirrhosis, this can progress to hepatocellular carcinoma. Now, why would it progress to hepatocellular carcinoma? See, the normal architecture of the liver is permanently deranged, and there is still repeated injury and insult to the liver. So in an attempt to keep repairing itself beyond its ability, the liver becomes dysmorphic and it might develop cirrhosis. Okay, so this is the natural progression of the disease. You start from a healthy liver, then you progress to NAFLD, then you progress to NASH. At any stage after NASH, you can progress directly to hepatocellular carcinoma. That depends on a number of factors. Now, there are multiple percentages that you see between different stages. Now, different researchers disagree on the percentage of progression from one stage to the other, but what they all agree on is that these stages do progress on to the next one in the absence of lifestyle changes or in the absence of intervention. So somebody with a normal liver, when they have NAFLD, they are going to develop NASH, they are going to develop cirrhosis, and they are going to develop hepatocellular carcinoma if they do not remove the source of insult and injury to the liver. Okay, so let's move on to our next slide. Okay, coming to my paper, reference intervals. This is a brief introduction to reference intervals. So reference intervals are basically health-associated benchmarks, and they enable clinicians to confidently interpret test results as normal or abnormal, and are therefore critical for accurately diagnosing diseases. So if you did not have a reference interval for an annulete, how would you know if the patient has a disease or not? So they're essential for making a clinical diagnosis, therapeutic management decisions, or any other physiological assessment. They're supposed to be established or verified for each annulete and specimen source in every country and in every lab, ideally, to achieve precise clinical assessment. So it is very important for the lab to carefully establish reference intervals by the laboratory according to standard protocols, for example, the CLSI guidelines. Now, if you want to study more about reference intervals, I just suggest you pick up TEATS or you pick up another textbook, because this is not going to cover all of it. This is just a brief introduction, okay? So this is a diagram showing the distribution of any annulete in the population. And what it shows, this is what you call a Gaussian curve. So what it shows you is that any annulete that is normally distributed in the population is going to show this distribution, which means that any annulete will be covered. 95% of it will be covered if you choose the mean value plus minus 2 st. Okay. So for any marker to be used in a clinical setting, a reference interval should be available for the relevant population, and it will allow the clinician to interpret those results with a certain level of confidence. It will also allow him to make an accurate and early diagnosis. So these reference intervals are utilized for diagnosis, monitoring, prognosis of disease, also for medical and surgical interventions and decisions. The best example for this is diabetes. Okay. So ideally, all labs should establish their reference intervals for annulates. It is also important for the lab to carefully establish reference intervals according to standard protocols, for example, the CLSI guidelines. Again, if you want to read up more on the CLSI guidelines, please read the description and links given in my video. However, to our knowledge, the reference intervals of chitinase 3-like 1 protein in diagnosing liver fibrosis have not been analyzed in our population, and our lab sought to validate and verify a novel biomarker for diagnosing fibrosis. Our interest in this marker was that this has the potential to be a non-invasive way of diagnosing liver fibrosis. So we sought to determine its reference intervals in our population. So there was no data for it in our population, both in health and disease, and these measurements would help us determine the reference range of serum chitinase in healthy people and determine its cutoff in patients with hepatic fibrosis. So coming to the literature review that I did for this paper, I found out other reference intervals or papers that have suggested it in different populations. So the kit literature suggested a cutoff of less than 79 nanogram per ml. This is from the kit literature. There was another paper that showed it to be 10.5 to 190 nanogram per ml in Egyptians. So you can see the kit, which is from China, says less than 79, but the study, which is from Egyptians, say it is 10.5 to 190. So there are obviously ethnic differences. So it is better to establish a reference interval in all different ethnic groups. Okay. There was one of the study from Pakistan. It was not a reference interval, but they were determining cutoffs for diagnosis. It was basically a radiological study, and they correlated it with this marker. It wasn't a reference interval study per se, but they calculated it as 90 nanogram per ml using ROC curve analysis. And then there was a Japanese study. it calculated the cutoff for significant fibrosis as 165. So you can see the wide distribution of the cutoff for significant fibrosis in different populations, and this just enhances the importance of determining reference interval in different ethnicities. So coming to my paper, so it is an upcoming marker for the diagnosis of liver fibrosis. The best thing about it is that it is non-invasive and it can be used to diagnose liver fibrosis. Our study aimed to determine reference intervals in our population and to determine its cutoff value for hepatic fibrosis. So this is our research performer. There were two research performers. One was for the healthy individuals and the other was for diseased individuals. So this is for the healthy individuals. And this is based on CLSI guidelines, the questions that they recommend, and you can also find that form CLSI document in the description of this video. And this is a research performer that we used for liver fibrosis cases. So it was a cross-sectional study. It was carried out in 2021, 15th January to 31st March 2021. There were a total of 408 samples, and there were 202 healthy individuals, and there were 206 diagnosed cases of chronic hepatitis, which we took for our fibrosis cases. So the kit that we used was an ELISA kit from a Chinese company, which is a sandwich immunoassay for ICT. And the statistical analysis that we undertook was done on SPSS23 and NCCS software, which I really love. I'm going to share the links of these softwares also in my description of the video, so that if you want to know more about these softwares, or you want to learn about them, or you want to download them for your research paper, then you can do so. Okay, let's move on to the next slide. Excuse me. So this shows you the distribution of chitinase 3-like protein 1 in healthy individuals. See? So you can see that this is a pretty good distribution. I wouldn't call it a normal distribution, but this is a pretty good distribution. These are our results that we calculated via our software via the percentile method. So we determined our reference intervals by using the interval covering the 2.5th to the 97.5th percentile in healthy individuals. And it turned out to be 12.8 to 81.8 nanogram per ml. I'm going to share the link of the software in the description. If you want to know more about the software, it really helps. Okay, this is the distribution in the hepatic fibrosis cases. So you can see this has a very poor distribution. When you compare it to the healthy individuals, this is a very poor distribution, and we had to use a lot of statistical measures to find out our reference intervals via the percentile method. Okay, so coming to the results of the cases, hepatic fibrosis cases, the cutoff that we calculated was 102.12, and it was calculated by a working normal method. So as the data you saw in the graphs before was not normally distributed. So we did a Box-Cox transformation procedure to bring the distribution closer to normal before we could calculate the cutoff. So what is a Box-Cox transformation? Again, I will put the link of the software in my description, and that software has a whole section of a description of the different statistical tests that it offers and when to use them and what is the procedure of those statistical tests if you want to learn more about them. Okay, so moving on to the next slide. This article has already been published a while back, and I'll be sharing its link in the description box below if you want to read more about it. I'll also be sharing all the different diagrams that I've added in this presentation. They're from different research papers. I'll also share any important links that if you want to read further on this, and let me know what kind of videos you want to watch next on my channel. Thank you for your time. See you next time.